Okay, so here's what you do. You go into Versum, and I'll show you in a minute what Versum is. It's one of the coolest things you've ever seen. And this is what you do. You take this nomenclature and type it in. So let me tell you what this is. Standard nomenclature. cDNA, that's what the C stands for, period. 641 is the nucleotide in the messenger of the cDNA from the messenger. So it's the 641st nucleotide. So you type in C.641, and that changed in this patient from a T, which it is supposed to be, to a C. So type that into Versum, right in that little search box. And it'll probably ask you to read a whole bunch of things like, within the first one.
Is that part one?
Huh?
Is that part?
Yep.
Okay.
Oh, no, I'm sorry. No. Hold it.
Okay.
Type in NF2, you have to say the G. NF2 space C.641 T greater than C, which is T changing to C. Now, let me see if I can do this with you. Did that work for people? Okay, hold on. All right. So NF2, can somebody read that to me off their computer? C dot. C.641 TC. Did I see enough one or NF2?
Yes, I get the same.
You go select all and confirm. And I did something wrong, but I can't see what it is. 641 TC. The reason that this is an important exercise is because if you order genetic testing on tumors or blood, you're gonna run into this. So go to this transcript right here, 268. Hold on. Did other people hit this? I'm sorry.
Yes.
Yeah.
It's HR 22, position 300. 3005.
Let me see. Yeah. Can I plug this in? Can I plug this in so people can see it in case they're hitting that same screen? I don't think I have that online, do I? That's the old one. Okay. So this is what it would look like if we could get you easily through that thing. Can you make it show the screen? Okay. All right. So this is the result that you get back. Okay. So there's our mutation or our alteration up at the top, and look at some of the kinds of information that this gives you. Can I make this full screen? Is this like that? Yeah. So look at the information that this gives you. I'll give you a couple of really cool examples, okay? And I'm sorry I couldn't get it to work on your computers, but one of the things that happens is that it goes through all of the gene databases where mutations have been reported and it looks for them. And so when I looked at this particular patient's mutation, it had never been reported anywhere. Here's so here's another another part of the analysis. So basically what it's doing is it's taking multiple genetic types of analyses in one web page and it's putting them together. So this says it's a missense mutation. Yeah, fair enough, we kind of knew that. And then it gives you an analysis of frogs, snakes, dogs, and asks whether the amino acid change that resulted from that mutation is ever been reported before, or is it always the same? And that's in this. So that T changes, you see, and it says that it's not that it's a conserved amino acid, meaning that it's always the same in a wide variety of different animals, suggesting that the protein gets screwed up if that amino acid changes. And then there are many different kinds of so-called in silico modeling systems that look at the known structure of a protein and ask whether the mutation should be damaging or not. And the cool thing about this Varsum site is that it looks at all of those and it tells you whether it's pathogenic or not. And so the vast majority, even though this change in this patient of mine was classified as unknown significance, when you actually go in and do the analysis in silico, that means in a silicone chip, you see that most of the programs predict that that should screw up the protein. And so basically what happens then is that it allows, what this program does is it allows you to apply a set of rules that look like this. And I want to give you a question. I want to give you the rules and we're going to try to apply them. But then we'll have to do it. Okay, never mind. I got it on the computer. So there's a set of rules defined by the American College of Medical Genetics that say all of these things that we just looked at, is it predicted to be changing the protein? Those things are all listed in this table. And now can I go, Megan, can I go back to my slides now, please? And that table looks like this. So, just look at some of the characteristics of this. And I did have this part out for you guys. So, is it located in a mutational hotspot or someplace that's critical for the protein? Yes. Is it absent from large databases? Yes. The mutation's never been reported before. If it's 50% of us have that information, it's probably not important. Is it assumed de novo? Well, in this case, we know that his family history was very clean, so yes, it's assumed de novo. And these are rules that come out of this Varsum site. Multiple lines of computational evidence to say it's deleterious. This is like protein conformation projections to say it screws the protein up. Yes, for sure it is. The patient's phenotype is suggestive of the syndrome. Well, that was the thing that got us started on this, because this guy got NF2, clearly has it, and there's no question he has the syndrome. And how is it that we just happen to pop up with an NF2 gene change? And so then what happens in this ACMG rule is that you take the various things that you've done about the gene from the Varsum analysis and then you apply it to this, and it turns out that that's a likely pathogenic mutation in this patient. Okay? And so what happens then is that you go, all right, I've got a patient with this, and all of the worldwide databases say that it's variant of unknown significance, but I say it's not. And you submit that new data to a place called ClinVar. So I did that because now I'm absolutely convinced that this is a disease-causing mutation. And sure enough, after their big committee went and looked at this, the Swedish Neuroprognosis Society, they reclassified that mutation or that alteration from a variant of unknown significance to likely pathogenic. And so now that is in some change like that night from the US to likely pathogenic. So this is really important because it allows us to do a bunch of things. This guy, remember, he only had hearing loss 4 years ago, so he's late onset. And so maybe his 50-year-old sister might have this as well. We just don't know. We don't think that it was present in her, but we don't really know about him or dad. And so we've already tested this row of family members and his mother, and they're all negative, except for him. He has the mutation, and they don't. And so as a result of that, it's a de novo mutation in this guy.
Can you just talk about what you said to ClinVar to?
Yeah, it's a wizard, so there's a standardized format, and you say, this particular gene, this particular C dot whatever mutation that we did, I have a patient that has a de novo case, it's clearly in F2. The in silico analysis says it's damaging, this says it's damaging that, and then I just took those rules. I took this information here, and I put all of those highlighted areas into the submission, and about 3 weeks later, it suddenly changed to a likely pathogenic mutation.
You have to submit it. You cannot go and go over imaging or the.
Not imaging, but I said 58-year-old man with bilateral vestibular schwannomas and a negative family history. That was the clinical information. And then I put that in there. And they said, terrific. Likely pathogenic mutation. And so it's kind of fun to do this because, it's really developing now. Anytime another patient is found to have that particular alteration, they'll be good to go. All right, so I'm sorry that didn't work quite as smoothly as I hoped it to. I hope it's still enlightening about how we analyze mutations because you get a lot, when you do genetic testing in the tumor setting, you get a lot of variants of unknown significance, and you have to think through whether it makes sense or not. And that's how you do it. So I've got 2 cases, and then do you have more cases? Or yeah, and I know Ricky's got at least 1 case. Yeah. And we've got 40 minutes? We've got about an hour to go. Yep. So let me just roll into a couple of cases. And as you can see, here's this mutation analysis again that we were just talking about. I'm not sure how to make that full screen. So, this is a really, really cool case. This young man came in to see Grace in the, I want to say 6 months ago. 28-year-old in a wheelchair. And what had happened to him is that he developed some symptoms, I can't remember, back in May of 18. And he developed some neurologic symptoms, gait difficulty, posterior headache, etc., and had this lesion. And somebody actually went in and biopsied that. They didn't try to resect it, but they biopsied it, and it was thought to be a pilocytic astrocytoma. It had low-grade renal features, it had some pilocytic changes in it. And there was a note in the hospital that he had come from that was scattered positive staining for BRAF V600E. He got radiation treatment without Timinar, and within months he was getting sick again. When he got to us, he was barely able to walk. And he was thrown up, and he was, all the things that you would expect from a cervical medullary junction lesion. And what happened to his tumor after radiation treatment is it went from here to here. This is what we saw back in the imaging. And so he clearly had progression despite radiation treatment of this presumed pilocytic astrocytoma. And he moved to the Seattle area and became our patient. And so we went through his records and noticed that there was this issue about a possible V600E mutation. Now, the way V600E works, BRAF is an oncogene, and this means that the DNA alteration changes the valine at 600, the position 600 to E. Everybody is the E. Okay, and so this is the amino acid that gets changed from this to this, and when that happens, and I think Santosh already mentioned this in the case, when this happens, it drives cells to become very malignant, and melanoma is the classic place where this BRAF mutation is seen. But sure enough, pilocytic astrocytomas often have the most common genetic change in pilocytic astrocytomas is a BRAF mutation. And the thing about it is remember that discussion around a driver mutation, something that kind of starts the tumor cell, starts the cell off down the pathway? This is one of those changes. It's the first change that happens in this guy's, one of his cells in his brainstem, and every tumor cell in that tumor ought to have a V600E mutation. It's a driver mutation. We don't see this typically as something that kind of arises as a tumor gets more malignant. So remember that I said that if you can tackle the driver mutation, you're more likely to be successful at treating the tumor. Okay? So the thing about staining for BRAF is that it's not a very robust way of detecting mutation. Most people would rather have a mutation analysis at the DNA level. So I got his tumor tissue, sent it off to Tempus, and got back a missense gain of function mutation at V600E. So sure enough, his tumor has got this, and we didn't send one because I didn't think he had a syndrome. He's got one really, really interesting other genetic change here as well, but that's for next time talking. So he's got, now he's got a protein change that was subtle, and he's clearly got this missense mutation in his tumor. So here's the deal with this. What happens is that there is predominantly sort of EGFR-driven signaling in the cell, and one of the things that can drive this signaling pathway to tell cells to divide rapidly is a BRAF mutation, V600E. And we have drugs that can block BRAF and V600E. It's a small molecule pill. You take it, it knocks out the function of this oncogene. And then it turns out that if you do that, there's a reflex pathway which drives other parts of this pathway to be even more active. So if you use a BRAF inhibitor by itself, the tumor only responds briefly and a little bit, and then sometimes it can actually begin to grow more rapidly because of bypassing pathways. So what the melanoma people discovered a long time ago is that what you do is that you add in a MEK inhibitor, which now gets around some of this pathway being driven by the BRAF. And if you do this so-called dual MEK inhibition, you can take somebody with extremely dense burden of melanoma, and 2 weeks later, all their skin lesions go away, and their lung lesions go away, and whatnot. So we said, he's got a V600E mutation. Let's give him a dual MEK inhibitor. You ready for this? This is crazy. So here's his tumor getting worse. He's in a wheelchair. November, I didn't put that. We have a more recent scan than this, which is even better than this one. November, he's playing basketball with his friends and taking 2 pills a day. It's a miracle. It's an absolute miracle. I mean, just look at this. It's crazy. And this is because we got the driver mutation in his tumor, and we're able to kind of, cut the tumor off at its very root, if you will, and then all the arms of the tumor die. So, a little bit of side effects. It took us a long time to get the ability to give him those 2 medicines, but it is, this is what he's getting. He gets it every day, 28-day cycle. Counts, liver function tests, et cetera, doing fine. But that's what you can do with these really powerful mutation changes. And any of you who regularly treat brain tumor patients in the room have seen this kind of thing. Santosh had a case as well. But it all depends on being able to do this genetic analysis and understanding what these these alterations mean. There are a lot of other BRAF alterations in tumors, and if you give them, if it's not D600E, but it's something else, it can drive the tumor. And if you give them dual MEK inhibitors, you can actually accelerate the growth of the tumor if you use it for a BRAF mutation that's not that. So you got to know which mutation you're going to target. So any discussion about this? I mean, this is, you know, if we could only do this with glioblastoma on a regular basis, how much fun would that be, right? It's possible. I actually got a BRAF glioblastoma the other day, which is it's going to do the same thing.
So you're just doing an off-label use of the drug. You're not doing an IND.
Right.
And you have this vehicle now to have this feedback to but what happens so that others will be aware of this data for this outcome? I mean, it's fantastic.
It is fantastic, and this, so it doesn't sound like I discovered something. This is pretty, I mean, this is, I don't know how to get myself out of this at this point. This is pretty well known.
But I mean, other people that want to do the same thing, it's not going to be published.
Well, what I'm saying is that there's quite a bit of work now in BRAF, V600 brainformers of various kinds, that if you're into that literature, like, he wouldn't miss it, right? But, you know, I almost missed it because I wasn't sure I'd going to be in my histochemistry result, right? I go, I better send off for a mutation, right? And I'll tell you that 4 years ago, before I started doing a lot of genetic analysis, I would have just said, fine, so it's kind of
How does this get to the FDA and get on label eventually? I mean, does it mean it has to have a clinical trial? And
no, I don't. I mean, Rick, you're saying you could apply on this. I don't think it could never be approved for pilocytic astrocytoma. It's always been off label because it's such a bloody rare disease, right? So I don't foresee that there would ever be an FDA approval process for it, but we have no problem with fully putting people on this.
it just seems like with each of these discoveries, it's going to be more and more often that this comes about with more and more molecules. Well, yeah, so that you're going to have to invent a new pathway and get to the FDA and get it on label.
Yeah, I mean, it's a challenge because I think, you know, in medicine we do a lot of off-label treatments. The publications do help, though, whatever case reports or case series, because when we submit the approval for the insurance, we back it up with this data, and that's helpful. But John's right, this is known for several years, that this does help gliomas, and that's the information that we submit. But it never hurts to publish more either.
But I also think I think one of the points that you're kind of driving at is how do we make more people aware of this?
Yeah.
Because I will tell you, as a neuro-oncologist in the field for 25 years, I have not paid attention to that histochemistry report 3 or 4 years ago. So now I'm sensitized to it. And, you know, I don't think we're going to cure this kid, but it's as close as we get.
There are
Any points on that? I mean,
there like the database where you updated the knowledge base for others to look at. There are people and databases that are trying to actually, in terms of providence and EPIC and genomic information, people are trying to put outcomes in there. In fact, many of the companies that do this testing want clinical data back so they can make these connections with what the clinician does with that information, what drugs they gave, and what was the outcome. And then so far.
Well, the outcome is very convincing.
it's kind of like penicillin, you know. It's you don't have to do a clinical trial to show that that's important. I mean, the kid's up and around, and so I'm going to show you one more, and then we'll go to Sam.
John, can I add a thought to that?
Where's the? Oh yeah, really.
So, no, I think it is still a clinical issue because I've had patients who had BRAF mutations that we were trying to get the drug for, and they've been denied. And you know, I show them the case series, try to really cite the information until we have phase 3 trials, which we will probably not get in such a rare disease. It still becomes a hurdle that I think limits everyday practice. So it is a good point to what other mechanisms can we do to effectively convince the FDA in these rare situations in this particular disease group to then provide this therapy as more standard.
So, as I mentioned, I think Santosh and I both mentioned, this was actually learned in melanoma. And check this out. Here's a patient with multiple brain metastases from melanoma. Goes on dual MEK inhibitor therapy because her melanoma has the V600E mutation. And this is no radiation treatment, okay? Goes from that to that. That's just revolutionary. This is crazy. All right, one more quick one. Non-small cell lung cancer, a woman from up north, a couple towns north of Seattle, comes in and has some neurologic symptoms and non-small cell lung cancer with this MRI scan. So, like Jerome was talking about this morning, there's two things going on here. Multiple peripheral metastases, and then that's easily recognizable as pretty bad meningeal cancer. And we scanned her spine, and she had a lot of enhancement in her spine. It didn't actually tap her, which, as I'll talk about in a minute, was a mistake. But proved that she had meningeal disease, but she's got meningeal disease. And anybody disagrees? Nobody disagrees with it, right? So it turns out that her lung cancer has an EGFR mutation in it, which activates the EGFR gene. And it's one of the known mutations which can be targeted by EGFR, oral EGFR inhibitor. And she had actually gotten back early, she had gotten Erlotinib and had a nice response in her lung. And then, because her lung responded so well, they took her lung out, and she had terrible infectious complications after surgery, and was off treatment because of all these things. Got better, everybody was happy with how she was doing, and then she comes in like this. But she still has this mutation. Now, if I was smart, what I would have done is I would have done a spinal tap, gotten her tumor cells, sent it to prove that the leptomeningeal disease still had that mutation if I was smart to do that. However, we put her on Osimertinib, which is a third-generation EGFR inhibitor, because of this, at double dose. So I can't remember who was talking about, Machi, I think it was, no, who was talking about how you get a jazz the dose up to be able to really get it in. I think anyway, we gave her double dose, and what we're doing is what we're doing here is we're tackling that mutation right there with Osimertinib, small molecule, oral, 80 milligrams a day. We gave her 160 milligrams a day. Big study just a couple of months ago in JCO showing how effective this is. Check this out. I've never done this before. She goes from this on pills to that. Sorry, to this. So this is her, this fourth ventricular region is now gone. The leptomeningeal enhancement is gone, and her parenchymal brain metastases are now scars. Pills. No radiation. It's extremely exciting stuff. Now, that's 5% of adenocarcinomas that have that, so it's not a daily occurrence. The thing that's is that the people that get this are typically carcinogen lung cancer. It's typically younger patients who never smoke. It's more common. You know, it's adenocarcinoma of the lung, in which case they respond extremely well. All right, so that's my second case, and Santosh, if you got a, or you want to, Ricky, you want to?